Okay. Okay, guys. So today we're gonna go over the major themes of anatomy and physiology in this unknown college, this mysterious college that nobody gonna know about. Okay, so we're gonna study the difference between anatomy and physiology. So anatomy is the structure, physiology is the function. Anatomy and physiology are inseparable. The anatomy is dependent on the physiology, and the physiology is dependent on the anatomy. A knife is sharp because it's able to cut. A hammer is dull. It can hammer things down. Both of them are made up from steel, but their structures are different because their jobs are different. So a cardiac myocyte, a cell of the heart, a skeletal muscle, a cell of the skeletal muscle system, a neuron, a cell of the nervous system, and a hepatocyte, a cell of the liver, and so on. They all have the same DNA, but do they have the same structure? They do not. Histologically, they have different structures because they have different functions. So the skeletal muscles have a lot to do with mechanical activity. So they're going to be full of actin proteins, myosin proteins. The heart is going to be full of, these cytoskeletal proteins that help it pump the blood. But the liver cell does not need to pump the blood, so it's not going to have as much cytoskeletal proteins. Okay. Alrighty. Okay, so anatomy is the study of the structure of the human body. So we begin with inspection. So we look at what's wrong. Is there redness? Is there an injury? Is there swelling? Is that part swollen? And then there's palpation. Palpation is when you put the palm and you feel the sensation, like when you palpate for the pulse, okay, the radial pulse. And then there's auscultation. Auscultation is when we use the stethoscope, and the stethoscope we hear the sounds of the heart, the heartbeat. We hear the sound of the valves. Okay, is there a murmur? Are the valves too stiff and blood is unable to pass through from one chamber into the other chamber of the heart? We hear the sounds of the lung. Is there tactile vocal fremitus? Is there some sort of is there some sort of lung fibrosis, or are there changes in the breathing sound? Percussion is when we tap on the body to feel the resistance. That's how we tell that our organs got increased in size. Pathological increase in the heart, in the liver size. Pathological increase in the liver size known as hepatomegaly. Pathological increase in the spleen size known as splenomegaly. Then there's auscultation. Auscultation is listening to sounds produced by the body. We talked about that. Dissection is when we cut and separate the human body tissues to reveal the tissue relationships. We use a cadaver for that, a dead human body. And we're going to see evolution happen. There is progress happen. And the progress is back in the day, they used to do dissections on slave. They used to do experiments on prisoners, but then they realized this was unethical, so they abolished completely any experimentation whatsoever on human beings, whether alive or dead. Then they realized that the alternative for that is animals. We can do research on the animals until they realized in the end that the animals are different from the human being. That's a comparative anatomy. They're not the same. The liver of the monkey does not look just like the liver of the human being. It looks like four finger projections. So now They went from thinking that if you take the blood of the lion and you drink the blood of the lion, that will make you strong like the lion. They realize that's all wrong now. You know, some tribes in parts of the world would drink the blood of the animal, of the lion, thinking that if you drink it, it's gonna make you stronger. Yuck. And then there is comparative, but they don't know. They think that's how it is. And then there is exploratory surgery. That is when they open up. If you say I have abdominal pain, they say, well, then let's see what's in there. Let's open up your belly to see what's wrong inside. Then they realize, no, radiology is the best thing. We use diagnostics. We use ultrasound, X-ray, CT scan. That helps us see what's inside. Okay, subdisciplines of anatomy include gross anatomy, that is the study of the structures that can be seen with the naked eye. Then there's histology, and that's the microscopic anatomy that where we examine the tissue. So they went on from thinking that anatomy is a holistic field where we look at the general body structure. I'm gonna forward you guys this lecture PowerPoint so you can just relax and enjoy the lecture and focus on learning than to take a snapshot, making your uh, album, your photo album, like your using your memory, your gigabytes. Just pay attention and that's all that matters. Histopathology is looking at the mechanism of action for mechanism of disease spread. How does the disease happen? You have cellular stress that later on becomes cell injury that leads to cell death, that leads to cell inflammation, and that leads to tissue inflammation, acute inflammation, and chronic inflammation. Then there is cytology, and that is when we focus on the cell and below. We talked about that last week. We said that we have some problems that are on the systematic level, signs and symptoms on the organ system level, but we see that it was caused at the level of the central dogma of biology. These antiretroviral medications. These antiretroviral medications are not happening at the level of the cell. They are working with enzymes and proteins that are happening subcellularly. Okay, physiology is different from anatomy. Physiology is using the methods of experimental science, where there are subdisciplines of physiology, such as the neurophysiology, and that is the physiology of the nervous system. Then there is endocrinology, and that's the physiology of the hormones. And then there's pathophysiology, and these are the mechanisms of disease. So we study how disease forms. And then we have comparative physiology, and that is we study different species, and that's how they were able to find out that the human beings and the chimpanzees, as far as DNA is concerned, we have a very close lineage. So the human being is closer to the chimpanzee than the chimpanzee is closer to the gorilla. We and the chimpanzees are closer to each other than the chimpanzees are closer to the gorilla. Why is that useful? That is useful because we can use the monkeys as animal models. We can test the medications on them. So medications tested on monkeys will give you results that are closest as if they were on a human being. Then we learn that in evolution, people think that the evolution theory says we evolved from monkeys, that they were monkeys and we came down from monkeys. This is a gross misunderstanding of the theory of evolution. Did evolution say that we came from monkeys? Did the evolution say that? Okay. Did the theory say that in your family photo album, when you look at a picture, you say to your mom, who is that? That's your grandfather. Who is that? That's your great grandfather. Who's that? That's your great grandfather. Who's that monkey? That's your great granddad. So it doesn't work that way. We didn't come down from monkeys. It never said that. It's the people who don't know the science, they always make they make rumors. So what did it say then? It said that we happen to have DNA close to the monkeys. That's all that it says. Okay. Okay, so now that shows you the history of anatomy and physiology. And that began with Hippocrates. Hippocrates said that, let us attribute disease to its natural causes. Okay, back in the day, they used to say, you are sick because the demons and you are possessed by the devil, and all of the demons are angry with you, and all that kind of stuff. So Hippocrates said that no, let us look for the natural testable causes of disease, like virus. Was the disease caused by an infectious microbe? Was it caused by a virus? Was it caused by a parasite? Was it caused by a worm? Was it caused by a form of fungi? And was it caused by bacteria? So he said, let's look for natural causes. Was disease caused by some chemical agent, a detergent we spill on the skin that caused some tissue irritation? Was the disease caused by some physical agents like a real high temperature or a really low temperature, and so on? Then Aristotle believed that diseases had supernatural or physical causes. Even if that's true, it may not be entirely wrong. Maybe some diseases are caused by some demon or stuff like that. But if it's not testable, we cannot run it on science. So when it comes to medicine, we only deal with science because we can test the effects of salt on blood pressure. Because I have salt, I have a heart, and I have a blood pressure, a sphygmomanometer. I can use it to test my blood pressure. But I cannot test the effect of the devil on the heart rate. Okay, because I cannot test the devil. Maybe the devil is there. We're not talking about if the devil is there or not. Maybe it's there. But all I'm saying is if I can't test it, then that is not science. So we should just focus on what we can test and try to repair that. Then Metrodora came in, and that was the first woman, the first female to publish a medical textbook, and it was about obstetrics and gynecology. So in obstetrics and gynecology, she was talking about the diseases that influence and affect females. Then Claudius Galen came in, and that was a Roman doctor who was dealing with the gladiators. You know, the gladiators for entertainment, they punch each other in the face and is bleeding and pus. So he would collect that and study it. And actually, he put in his encyclopedia a lot of information. A lot of it is wrong, but some of it holds true up to today. So we take the true and we leave the rest. But his effort was okay. So the more work you do, the more right and the more wrong. So the more work you do, the more you're going to be able to produce to the more you're going to be able to like get get the results. Okay, Maimonides, that was a Jewish physician. So the Jewish physicians and Muslim physicians were able to contribute a lot to science in a time where the church disallowed scientific research. They used to think back in the day that disease is from God, and the church would ban any form of experimentation on human beings. But that was not happening in the Jewish and the Muslim world, so they did that. But in the end, they combined all the information, and it all became part of the science that's today. So medicine, in its essence, is an art. You have to have a talent in the way that you examine. You examine the problem and how you are able to repair it by looking at the problem from all the sides, not just from one side. Okay, so now that was the end of old medicine. Now there is a new modern era of medicine, and that's where Andreas Vesalius came in, and that is at the time where they relaxed the restrictions on on research. But when they allowed it, that if somebody died, you can do dissection on them. They didn't know embalming. You know the embalming and removal of blood and putting all the formaldehyde to preserve the cadaver. They didn't know that. So the cadaver, oof, was smelling horrible. So now everybody, imagine all of us are in a lab learning anatomy, and there's a dead person smelling very terrible. You can smell them from the end of the block, but you have to be really close so you can learn what's in there. And then there was a barber surgeon, the person who used to get really close and open up and dissect and all of that. So Andreas Vesalius actually came in, and the anatomist would used to sit at a very high chair known as the cathedral. But then Vesalius came down, and he was actually with them in that stinking environment to teach and to learn himself also. And then William Harvey came in. So Vesalius, came down from the cathedral to work on the anatomy and the physiology. And he published the first atlas of anatomy. I mean, if you're ever asked who published the first atlas of anatomy, it was Andreas Vesalius. Then William Harvey came in, and he published a book, and he was really concerned with blood vessels. When we get to that, when we get to blood vessels, we're going to be able to see what experiment that he did with the blood vessels in the hemodynamics. Then we talked about this in lab, and that is when there were developments in the microscope, and the microscope really helped us learn. And what happens was Galileo. Galileo was so concerned with what? We said in lab that Galileo was concerned with what? I'm sorry? Yeah, the telescope and what's happening in astronomy. Very good, in the skies and the stars. Then what happened was they said, why are we looking up? Why don't we look down into the petri dish? And why don't we see our cells? Maybe there's something that we need to find out in our own body. We're going out there in the galaxy to learn about what's happening in the astronomy, but we really don't know what's happening in our own body. So they made just like a telescope, but now it's a microscope. So we're not trying to see things that are far, we're trying to see things that are really small. But we use the same idea, which is a objective lens and an ocular lens. Birmalpigi came in, and he observed. He actually observed. He was the one who changed the telescope into a microscope, and he was able to see blood cells and capillaries. Then Robert Hooke came in, and he made us a microscope that could see 30 times as big. But that wasn't big enough. And that was Robert Hooke's microscope. Then Anthony van Leeuwenhoek, he was able to invent a microscope with a single lens, but it can show us 200 times. Now that was really big contribution in science. Now we can see a lot and we can discover more. Then Schleiden and Theodore Schwann. Schwann has the Schwann cells. We will learn about them when we are doing the anatomy and physiology of the neuron. And he concluded that all organisms are composed from cells, and come from other all the cells come from other cells. Yeah. Who said that? Very good. So it wasn't the demon, it was somebody who said that. Very good. Excellent. Cells come from other cells. Cells don't come from nowhere. So if a tissue is injured and there's necrotic tissue and inflammation and the cell died, that cell has to be removed, and the other cells will go in the cell cycle to burst out and birth and make more cells. So they didn't come from nowhere. They didn't come from the demon. They came from other cells. Very good. So there are multiple theories, and then there's the germ theory of disease, where we know that infectious diseases can cause, these infectious organs secreting endotoxin, exotoxins, poisons that degrade the integrity of our cell membrane and break our cells, and then all the content of the cell can spill into the tissue. Then there are mechanisms of heredity. There is heredity where there's autosomal genes and sex-linked genes. There are sex-linked diseases. There are autosomal diseases. There are dominant diseases, and there are also recessive diseases, and so on. Okay, then we move on to the scientific method. And we talked about that in lab. And that is, we have to have logical thinking, inductive and deductive reasoning, looking for the truth. Things that we can make a testable, falsifiable hypothesis. So a testable hypothesis is a hypothesis I can test, I can run. It's not something that is not there. I think about it, but it's not there. And then I can make it has to be falsifiable, where if this medication causes a disease, I'm sorry, if this medication brings down pain, I will falsify it by saying if you take the medication and it does not bring down the disease, the pain, that means the medication is ineffective. But I cannot go one route and say, if you take this medication, your pain will go down. And if you take it and it doesn't go down, then maybe it's not something wrong with the medication. Maybe it's something wrong with you. It doesn't work that way. You ensure that your medication is working. You can't just say, well, something is wrong with you. Okay, elements of experimental design. So we have to have a sample size, where the larger the sample size, the better. Would you like a medication that was tested on 1,000 people, or a medication that was tested on 100 people? 1,000 people. Why is that? I can't hear.
It's more accurate.
Yeah, why is it more accurate 1,000? Very good. It's more accurate. That's an observation. But what is the reason? Why is it more accurate with 1,000 than 100?
More research.
Yeah, because reliability is higher and validity is more valid. Validity is higher. Very good, very good. There's control groups. So I cannot test 100 people who are suffering from pain, that is level 10 out of 10, and give all of them my Tylenol and then assume that, and when Tylenol comes down, say it was brought down because of my medication. Well, you had no control group. But if you split them in 50-50, you give one group Tylenol, the other group you don't give Tylenol, and the group that received the Tylenol, their pain went down, and the group that didn't receive the Tylenol, their pain didn't go down. Now that makes sense. It was your medication. But if all of them, their medication went down and there was no control group, you can never assume. You can never assume that the effect was due to your medication. Okay? All right. Statistical testing. Statistical testing is very important because I have to find out how statistically significant my results are. I mean, if I take a medication and my pain goes down by 2%, my pain was 100. After I took the medication, my pain is now 98. And another medication, when you take it, your pain will become 50. And then another medication, when you take it, your pain will become only 2%. Which one is the best? The 2%, because it dropped so much. That shows you it's more statistically significant that your medication was able to bring down the pain 98%, while another medication, its pain brought the pain down only by 2%. There's experimental bias. This is a really important concept. In experimental bias, what happened is that sometimes the researcher is so eager to get results. It's very tedious to set up an experiment and to get the funding, and you are in a rush to get the results, so you interfere with the results. You jump the gun and you say Tylenol is a good analgesic. Well, you haven't run it yet. So you start making bias. So how do we solve experimental bias? By double blind method. What is the double blind method? In the double blind method is you have your three components: your researcher, the control group, and the experimental group. What is the difference between a control group and experimental group? Yeah, they don't take any treatment. Very good. Yeah, they take very good. Okay, is that clear? So if in the experimental in the if you receive what you are testing, if you receive the medical treatment that you are trying to test its effects, then you are part of the treatment group. If you do not receive the the what you are testing, the Tylenol, then that makes you control group. So the three groups, let's say this half of the room is going to receive the treatment. The other half is the control, and I am the tester. I am the experimenter. Double blind means I don't know which group is control, which group is treatment. And you don't know if you are part of the experimenter group or you are part of the control group. So that's double blind. If double blind is there, then we have eliminated completely the bias. Because I don't know, I'm not gonna go mess around with people and tell them you took you took the Tylenol, okay? So your pain should be going down, okay? So don't be saying nothing else. Okay, in about a few minutes, your pain should be going down. Your pain went down, right? Your pain went down. So that's what they were doing. Okay, so you are so eager to get results, and you are so now you're interfering with the experiment, and you go to them and say, "Listen, you guys over there, you're the control group. Okay, you're not going to be getting any pain medication. Okay, so don't be saying my pain went down. Doesn't make any sense. So when you do that, you have messed up the experiment. But when you don't know who is who, you say, okay, you guys, some of you are receiving the medication, some of you are not. So nobody knows who are they part of the group. So he's gonna tell them, okay, everybody take your medication. And he's like, okay, so we'll find out in the end. And that's how it's supposed to be. Okay. All right, guys. Peer review is important because you get counsel. Counsel is very important even in the science world. So people, now they make decision without getting counsel. They just rely on themselves and they're like, okay, if I have a problem, that's how I fix it. There's an advising center that you go to, and you don't just go over there and you do what you want. No, you have to go and you have to have faith in the person and trust them and do what they tell you because they are expert. But you don't go get counsel from the Facebook. They'll tell you, okay, take this pill if you want to lose weight. It's a peppermint and blah blah blah. You take it, it's gonna. No, that's not how it works. You have to go to the medical doctor, to the nutritionist to help you with that specific problem. But if I have a legal problem, I'm not going to go to the nutritionist. I'm sure he may have something to say about it, but I'm not going to take it from him. I'm going to take it from the lawyer. Okay. And if I have a stomach pain, I'm pretty sure the lawyer may tell me what I need to do to repair it, but I'm not going to take his word for it. I'm going to go to the doctor. Same thing with peer review. Before we embark on an experiment, we have to have a couple of other scientists reading it before that and telling us, well, that has been done before. It's cost effective if you change the experiment. Maybe you can test two things at the same time. Maybe. You know what I mean? You take counsel. So it's very important to have counsel. And it's very important to have, you work with your colleagues, no matter where you are. I'm a professor. I have colleagues who are professors. We have to work together. You are students. You have to work together as students. So it's very important that you guys are working together as colleagues, the same way scientists work as colleagues. Okay, facts. Facts are information that can be validated, verified. What's your phone number? My phone number is 12345. Take out my phone. 12345. If you pick up, then that's a fact. If I call 12345 and then he picks up instead of her, then that's not a fact. That was a lie. If it cannot be verified. The law describes one clear-cut thing. The law describes something. The law of motion. It's only described motion. But theory, it describes multiple things. the theory of evolution and natural selection. This and blah, blah, blah. So when multiple things, that's theory. Okay, so Charles Darwin, we talked about that. He wrote the nice book, The Origin of Species. And what did he say? He said two things. He said that we evolved from a common ancestor. We all developed. So he's saying that we came from a common ancestor and we will continue to change as long as there is selection pressure. So, for example, this is happening over years. People who live in the top of the mountain where there's a little bit of oxygen, they will tend to have compensatory red blood cells that are red blood cells that are they increase in number so that they can accommodate more oxygen. And then there's adaptation. These are inherited features that we get that help us deal with challenges, like the lizard, who get a better camouflage than the generation before. Sometimes it's really hard to get them. They look just like the leaves that they hide behind. And then there's and then there's selection pressure. And selection pressure is there is certain type of pressure that helps you to succeed and survive. So this predator who is faster than the other predator, that type, that form of those predators, they will survive. They will survive and move on. And the ones who are too slow to catch the prey, they will die. So the more you keep up with the selection pressures, the more you will survive. The less you keep up, then you will not. Okay, and these some adaptations are, being arboreal. Arboreal is when you are living on the top of the tree. Okay, then we develop our eyes. The old human beings, they had eyes that are on the side, like the fish and like the animal. But now we no longer have peripheral eyes. We have central eyes. We have better stereoscopic division because that helps us survive. If your eyes are here, you can see better, you can integrate information better, and you can take visual input better, and that helps you survive and see better. And then the color, you can see like depth of color better, so now you can tell which fruit is more ripe than the other one, and that's more delicious and more nutritious than the one that is less ripe. Bipedalism, being able to walk on 2 feet. We haven't been walking on 2 feet since the beginning. Millions of years took that, for us to be able to stand up and walk on 2 feet. And that was because walking on 2 feet helped us more than walking on 4 feet. So that shows you we are able to develop that those characteristics that help us survive. Okay, so now we are down to the hierarchy and the complexity. So we said that at the bottom we have molecules. Remember in lab we talked about how we have molecules and organelles that develop to giving us cells and tissues, and then we have organs that are composed of tissues and organ systems that are composed of organs and so on. So you see we have atoms that together they give us molecules, and molecules give us organelles, and organelles build up cells, and cells give us tissue, and the tissues give us organs. Then we have organ systems, and lastly, we have organisms. And many organisms give us a,
Organism.
so many organisms, so that is an organism, but many organisms give us population. Very good. Okay. So we said that an organism is a single complete individual. Below that is an organ system. Like I gave an example with the skin. It has epithelial tissue, nerve tissue, and you have blood vessels. So that shows you that you have multiple systems working together. And then you have an organ, which is a structure composed of two or more tissue types that work together to carry out a function. Then a tissue is or similar cells and cell product forming a discrete region of an organ and performs a specific function. Cells are the smallest unit to carry out all basic functions of life. Then an organelle is a structure within a cell that carry out a function. The molecules are particles composed of two or more atoms. Okay. Reductionism in when we go from up to bottom, when we dissect the organism down to organ systems, organs, and then tissues and cells. Holism is when we study the emergent properties that occur as we ascend. So now we're going the other way around. Reductionism is when we go from the biggest to the smallest, and holism is when we go from smallest up to the biggest part. No two humans are exactly alike. Even identical twins are different. So the anatomy and physiology we're going to be studying in this course is true for most of the people, but it does not mean everybody looks that particular way that we have learned. For example, normally your kidneys are supposed to look like that, but it doesn't mean that some people don't have pelvic kidneys. Some people have kidneys that are down here. Some people have kidneys that are connected like a horseshoe. Some people are normal, have normal aorta as this one, but here you see that different variations in the branching of the aorta. So now we talked about negative feedback and positive feedback and gradient. Remember, we talked about those three. And first of all, we're going to talk about the 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 properties. We can never define life, what life is, but we can reduce the definition of life to characteristics that differentiate a living person from a dead person. So, number one, organization. So, the living things compared to the dead or the non living, they exhibit a higher level of organization than non living things. And cellular composition. So, the living matter is always compartmentalized into one or more cells. Metabolism. So this metabolism is the sum of all internal chemical changes. There is also responsiveness or excitability. And excitability is the ability to sense and react to changes in the environment. Movement. The living are able to move. The dead are unable to move. The living are able to respond. The dead are unable to respond. Metabolism is the sum. So being able to perform metabolism, that means you are alive. Being unable to perform metabolism means you are dead. Also, homeostasis, being able to restore homeostasis means you're alive. Being unable to restore homeostasis means you are dead. The development. So we change in the form or is the change in form or function over time. And reproduction. Reproduction is when organisms produce copies of themselves and pass the genes to the offspring. Evolution is when we have genetic change from a generation to generation that occurs due to mutations. Okay. We said physiological variation. Okay. So let's say a patient who is 3 years old and they have a lung infection. They have bacterial infection of their lung. And then a young man who's 22 years old has a bacterial infection of the lung. And then a 95-year-old female who has an infection of the lung also. Now, will I be giving them the same medication to the baby and the adult and the really old 95-year-old? So that shows you we have to account for this different for this variation. So the reference man we're looking at is a 22-year-old, 154 pounds, light physical activity that consumes 2,800 kilocalories. Homeostasis is the ability to detect change, activate mechanisms that oppose it. We talked about that negative feedback and maintaining relatively stable internal conditions. And that was coined by Claude Bernard. Claude Bernard noted fairly constant internal conditions despite the external conditions like the temperature changing. And negative feedback allows for the dynamic equilibrium with a limited range around a set point. So there's a set point. The green range is the set point that we got to come back to no matter if things get more cold or more hot. So negative feedback is the mechanism that keeps a variable close to the set point, and because feedback mechanism alter the original changes that trigger them, they are called feedback loops. Okay, so if too warm, the skin blood vessels dilate, vasodilation, and sweating begins. So that's a heat losing mechanism. If it's too cold, the skin blood vessels constrict and the shivering begins. Okay, negative feedback in thermoregulation. So that shows you what happens in the room, which is very similar to what happens in our body. So the temperature falls to 19 degrees Celsius. So what is that? A stimulus or signal? So what is this? Our stimulus and signal here. So look at the whole pathway first before you answer the question. What happens in number one? Room temperature falls to 19 degrees Celsius. Two, the thermostat activates the furnace. Three, there's a heat output. Four, there's room temperature rising. Five, thermostat shutting the furnace. Six, the room cools down. Four, and then we stop the thermostat. So here, what was our signal or stimulus? Hypothermia. Excellent. Very good. Who is our sensor? The thermostat. Excellent. And what was our input signal? Temperature went down. Excellent. Who's our integrator? The thermostat. Yeah. It did both. Remember, just like the pancreas was able to detect, it was an integrator and an effector. Sometimes you can have both at the same time. So the thermostat activates the furnace. Then the furnace gives the heat. So what is the furnace now? Effector. So the one who did the effect. So the output came out from the thermostat to the furnace, but the furnace is the one who gave the heat to do the effect, the final effect. Very good. And then the heat output was the effect that came out of the effector. And then the room temperature rise back to 20 degrees Celsius, which is healthy room temperature, and that represents for us homeostasis. And then we shut down everything. Now look at the body. In the middle, set point. What's the set point? 37 degrees Celsius. Okay, that's homeostasis. If it went up, look, if it went up, that's 37 going up, 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 up. Is that hyperthermia or hypothermia? So now what happens? Vasodilation and sweating. These are the two effects, and the effectors are blood vessels and sweat gland. Who's the integrator? Who's the integrator who made vasodilation and sweating? Blood vessels are the effectors who dilated. The brain stem, that's for the heart. You're talking about the baroreceptor. Who? I told you it had a thermal nucleus. It had many nucleus, but one of them is thermal. It was the hypothalamus. Very good. Very good. Now, what about the opposite? What if temperature went down? If temperature went down from 37, we go down to 36.5. What happened is vasoconstriction. And then you start shivering. And then shivering thermogenesis. Excellent. We genocide or we make warmth with our temperature. Very good. Very good, very good, very good, very good, very good, very good, very good. Homeostasis and blood, the baroreceptor reflex, the baroreceptor, we talked about that. When we rise from bed, the blood drains from the head and blood pressure falls in this region. Detected by the baroreceptors that transmit signals to cardiac center of the brainstem. Then the cardiac center transmits the signals to the heart to increase the heart rate, raising the blood pressure and restoring the homeostasis. Okay. The baroreflex illustrates the common components of a feedback loop. So we have a receptor, and that was it sensed the change. We talked about that. Then an integrator, control center that controls the control center that processes the sensory information and makes a decision. And then the effector, cell or organ that carries out the final corrective action. The person rises from bed. You see what happens? There's no equality in the blood, so the blood drains from the upper body, creating homeostatic imbalance, and the baroreceptors above the heart respond to a drop in the blood temperature, and then the heartbeat accelerates in response to the cardiac center, and then the blood pressure rises to normal. Positive feedback is a self-amplifying cycle. It leads to greater change in the same direction, as opposed to the corrective action of the negative feedback. So we talked about that. It happens in childbirth, you see, because in childbirth, blood clotting, generation of nerve signals. We'll talk more about that, but also breast milk. Okay, but we're gonna get to all of those again. This is not the last time you're gonna see homeostasis, not the last time you see how we repair hypothermia, not the last time you will see how we repair how the positive feedback is gonna be happening over and over again. Even next semester, we're gonna be going on and on and on and on. Okay, so the positive feedback in childbirth. So the head of the fetus pushes against the cervix, and then the nerve impulses from the cervix are transmitted to the brain. Then the brain stimulates the pituitary gland to secrete oxytocin. Then oxytocin stimulates the uterine contractions and pushes the fetus toward the cervix. Gradient. So the gradient difference in chemical concentration chart. We talked about that. There's a thermal gradient. There is a gradient that is um, pressure gradient from an area of a high pressure to an area of a low pressure. Concentration gradient is when you have gradient from an area of a high concentration gradient of glucose to an area of a low concentration gradient. And there's electrochemical gradient when we combine concentration of atoms that are charged. So we have both. We have concentration of matter and electricity. So this is going downhill from an area of a high concentration to an area of a lower concentration. But we will learn that the sodium potassium ATPase is able to is able to go against the gradient. So usually if you have 500 people outside trying to come into the room, when you open the door, they're gonna come inside the room. But if you have somebody who, when you open the door, out of the 500 people, 10 walk in, and that person is able to grab the 10 and push them outside against the 500, and that's going uphill. So you will see the sodium potassium ATPase does that. There is a lot of sodium outside the cell, a little bit of sodium inside, and a lot of potassium inside, a little bit of potassium outside. So the natural movement is for sodium to go inside the cell from outside, and potassium to go outside from inside. The sodium potassium ATPase will do the opposite. If a little bit of sodium leaks in from outside, it will send it right back out. And if a little bit of potassium is sent outside from inside, it will throw it back in. And we will see why is that. So this shows us the pressure gradient. Right next to the heart, that's the highest pressure. Away from the heart, that's the lowest pressure. And then you would have the concentration gradient. A lot of glucose outside, a little bit of glucose inside. That's concentration gradient. And here you see electrochemical, where you have a lot of positive charged sodium outside trying to come in. And you see a little bit of negatively charged electrolyte inside that is trying to come out. So each one is pointing to the other direction. And then there is thermal gradient from your blood that is so hot to the cool air. So blood flowing from your warm blood into the air. Okay. So now this is we have finished. This is just showing us the how do we pronounce the muscles, and this is just talking about the radiology, and that kind of stuff is just general information. But the most important part is us learning about the homeostasis. Does anybody have any question? Okay. All right. Can you do me a favor and